Welcome back. In the previous lecture, I described tumor suppressor genes. In the lectures before, I described oncogenes. So the oncogenes are activated for cell division, and then inherited cancers and the tumor suppressor genes that resulted from studying them are inactivated for cell division. In this lecture, I want to describe the series of events that happens at the molecular level to give rise to cancer. And it really is the achievement of modern cancer research and the war on cancer. I'm going to describe how cancer develops as a series of molecular changes. And then I'll step back and describe more recently the Cancer Genome Project and how it aims to describe all these changes in tumors of individuals. Then we'll talk a little bit about genetic testing and involving the ethical and legal considerations of this field. So first, cancer developing as a series of events. You'll recall a multi-step model. The multi-step model describes the initiation and growth of cancer as a series of events at the organ, tissue, and cell levels. To reiterate, we start off with mutations happening from bringing new genes in, mutations due to chemicals, radiation, and we have an initiated cell. Now we can make more specific statements now about what those mutations are, of course. That cell then develops more mutations. We have a pre-lesion, and then a malignant tumor, and then a clinical cancer that gets larger, and a metastatic tumor. All mutations being accumulated. The achievement of modern molecular biology is that now we can describe each of these events at the DNA level. And hopefully, describing these processes will lead to specific targeted therapies. So it's worth a detailed look at this process. This is colorectal cancer. It's a great model for what I'm trying to explain in this lecture. And I will emphasize, I'll describe one other cancer, but I will emphasize that for many cancers, the events are specifically known as they are for colorectal cancer. Bert Vogelstein, shown in the photograph, is a physician scientist at Johns Hopkins University. And he and his colleagues decided to look at the development of colorectal cancer by taking specimens at each step along the development and examine oncogene and tumor suppressor gene sequences and look for mutations and changes in DNA expression. So here we have the path to colon cancer. This is the colon, and the lumen, or the central part of the colon where the food is here. The colon, you may recall, is the second last place, besides the rectum, where food is after digestion. Most nutrients have been absorbed at this point by the small intestine, and now the stuff is in the colon and it's being concentrated. We begin with a small lesion in step 1. You can see it as a very small red dot. That lesion begins to grow. It grows some more into a polyp. And the polyp is what can be seen in a colonoscopy. So in a typical colonoscopy, a physician will thread a camera with a little snipper on the end of it into the colon, and when a polyp is seen, they'll just snip it out, and they'll do tests on it. Next step, step 4, it's a full-fledged carcinoma. It's a cancer. It's now got mostly cancer cells. The carcinoma then grows to more advanced stages, and then you can get a stage 3 or 4 metastatic cancer that can spread to elsewhere in the body. Here's the achievement. Vogelstein and other people took samples from each of these steps and described specific genes being mutated at each step. So if you look at the graphic, the first step, the first gene that seems to be mutated is a gene which I call APC. We'll talk about what it does later on. The second gene mutated, now I want to emphasize for a second, the second gene mutated in this APC mutated cell. So imagine what this is. You've got a lot of dividing cells, and one of them picks up an APC mutation by spontaneous mutation or by carcinogen, whatever. Gets an APC mutation. And it reproduces. So you've got a whole bunch of reproducing cells that have APC mutations, and they do functional things in the colon. And then one day, an APC mutant cell develops a second mutation. look on the graphic, MLH1. And then a cell that has an APC and MLH1 develops a third, get it? And so now we will have six, in my graphic, 6 mutations that happen and accumulate in a single cell type. And when you have a whole tumor there with 6 mutations, you can get a metastatic tumor. Actually, there's more mutations than this, but I want to describe just the simplest model. So the first gene mutated is called APC. It's a tumor suppressor gene, not an oncogene. What does APC do? In normal cells, there is a transcription factor which is called beta-catenin. It's shown here. And APC binds it. Normally, beta-catenin will bind to promoters for cell division genes. The tumor suppressor, kind of like an example I showed you in the last lecture, binds up APC, right? And it not only binds it up and prevents it from going into the nucleus, and binds up catenin and prevents it from going into the nucleus and stimulating cell division, but it does something even worse. It targets that APC protein, that beta-catenin protein, that transcription factor, for destruction in a mechanism in the cell that's like a garbage disposal. Essentially grinds the thing up to bits. So terrific, okay? It stops the cell from dividing by stopping that beta-catenin transcription factor from getting into the nucleus. In a colon cancer cell, however, APC is mutated. And if it's mutated, it may be changed, the shape is changed. It doesn't bind to beta-catenin anymore. Beta-catenin then goes in, acts as a transcription factor, and you get cell division. So that's the normal situation. In people with inherited colon cancer, which is about 7% of the cases, they have a mutation in APC gene already, and you get a second mutation, of course, you can get full-blown cancer. So this is what happens in normal cells and in mutant cells for APC. The second gene, MLH. MLH stands for mutator L homolog. Don't worry what it is. MLH is a gene which is also a tumor suppressor gene. It prevents mutations from accumulating. So here is a normal DNA sequence and a mutated DNA sequence, and the MLH protein normally comes in and repairs this mismatch, a G and a T. MLH sees it, it says, oh, that's the wrong base, you dummy. Replace that with a T, you should have a C there. We all know A bonds with T and G bonds with C. So replace that, and so it puts the C in instead of the T. So it's very good for DNA repair. If MLH is mutated, we're going to allow mismatches to accumulate. And again, if they accumulate in genes that are involved in cell division, we could get cancer. The third gene mutated in this series of colorectal cancer is RAS. Well, we talked about this earlier on in the course. RAS is a protein involved in the response of signaling to growth factors. The growth factor knocks on the door, the receptor vibrates somehow, and RAS is one of the first players that gets stimulated. And then you get a whole cascade of dominoes falling, and the dominoes end with transcription of genes involved with cell division. If RAS is mutated, very often it's always on. It no longer needs external signals, and so it just pushes the dominoes down, and you get cell division being stimulated by transcription factors because RAS is mutated and is always on. And that's what happens in the colon cancer cell. The fourth gene that's mutated is called DCC. Stands for deleted in colon cancer. That's another word for mutation. It's deleted in colon cancer, first found in those cells. It's also a tumor suppressor gene. RAS, of course, is an oncogene, stimulates cell division. DCC is a tumor suppressor gene which normally codes for a cell adhesion molecule. Sticks cells together. In differentiation, that's good because the cells together in the lining of the intestine is going to be functional and the organ is going to be intact. In mutated DCC, you no longer have, the structure of this protein is changed, you no longer have the cells binding, and so it can form a tumor, and the tumor will be able to grow and separate from the wall of the colon. The fifth gene, P53, we just talked about it. It's the guardian of the genome, the Swiss Army knife that signals repair of DNA mutations, promotes apoptosis, programs cell death. If it's mutated, it's not going to do those things anymore. So cells will not do apoptosis if they get damaged, and they will accumulate genetic damage if they get damaged. They won't try to repair it. The sixth gene to be mutated on my list are anti-metastasis genes. Remember in a previous lecture I described the steps in metastasis. a cell, the tumor migrates toward the blood vessel, a cell separates, it gets into the blood, it travels in the blood, it stops at a new place, it gets out, it grows a metastasis. There are proteins involved in all of those steps, and a series of genes involved controls those proteins and prevents cells, they're tumor suppressors, prevents cells from doing metastasis, doing all those functions. It keeps them close to home. That's the normal situation. Metastasis is blocked. When these genes are mutated, of course, they will allow the metastasis genes to act, and the tumor will spread. So the path to colon cancer involves a series of mutations accumulating over time. Now, a single mutation event may get it started, like a person born with APC mutation. That's a kickstart. But you're going to need a bunch more mutations for it to happen. And that explains why tumors, even in people who have inherited colorectal cancer, take a long time to develop. You need a lot of mutations happening over time. And you'll recall lab experiments where I described you need more than one mutated gene to get cancer in many instances. Question, what causes these mutations? Think of the colon. Think of what's in the colon. There's a lot of bad stuff in there, that's some of which is going to cause mutations. Some of these genetic mutations can be changes in gene expression. We'll talk about that in a minute. DNA methylation turning genes on or off. So, the biography of the colon cell is one of accumulating mutations. A little bit of anatomy here. Here's the anatomy of the colon in cross-section. So this is the wall of the colon, and here's the food out here. Okay, so it's the wall of the colon. And I want to emphasize what we're talking about. At the base of the colon is a stem cell niche, where stem cells are dividing and dividing and dividing and forming more cells. Now, why would you want to keep doing that? Here's the reason. The cells that are formed here begin to migrate up the colon. And these are little fingers that give you more surface area. So if we had the colon just like this, we'd have only cells here, lined up here and here. If we have a bunch of little fingers lined with cells, we can have cells here. You see there's going to be a lot more surface area to absorb food, and that happens throughout the digestive system. So the cells are migrating up the colon. They specialize and differentiate here, and they will differentiate to absorb water in this case and some food. And cancer also develops in these cells. And then the cells typically will die at the very end. I've actually done research on this particular issue on colorectal cancer. And these cells, it's very interesting. They take about 72 hours to migrate. They get formed, they specialize, they do their thing for a couple of days, then they go to the top of the colon, and then they do apoptosis, programmed cell death. That's a good thing that's evolved that way because, remember, these cells are exposed to a lot of bad stuff in the digestive system. And so they're going to accumulate mutations pretty rapidly, so it's good that they have apoptosis. But if you lose that many cells of your gut every day, and there are estimates that you lose over 1 pound of cells a day just from your digestive system, you've got to replace them. Constant replacement by stem cells. And it's good that you lose those cells because they don't accumulate mutation. If you biopsy cells of the colon from young people, you'll see the couple of mutations already, of these 6 mutations or 8. And by 60 years old, you'll see 4 or 5. And then, of course, later on, if you get colorectal cancer, you'll see the whole group. Here's lung cancer, the pathway to lung cancer. It's my second example I'll describe. Lung cancer, the lung, anatomy of the lung. The lung has tubes, and the tubes end in sacs called alveoli. And so a lot of surface area, little balloons at the very end. And these balloons are lined with cells, of course, and blood vessels to receive the oxygen and release the carbon dioxide. Cigarette smoke lands there, and asbestos particles can land there, etc. Well, the lung cancer develops in stages as well. You begin with a normal lung tissue. It's got these air sacs, and then you can have precancerous cell divisions, and then lung carcinoma and invasive lung cancer, different steps. And so people have taken biopsies of all these steps, and they've done a molecular biography of lung cancer development. And it turns out there are several genes involved. A mutation in a P16 gene, RAS gene, P53. Let's go back a minute. P16, I talked about that in the last lecture. P16 is a tumor suppressor. Okay, it's a tumor suppressor gene. And when you get a mutation, it allows cell division. RAS is an oncogene. And when you get a mutation, it stimulates cell division. And P53 is a tumor suppressor gene. and when you mutate, you get allowing cell division, right? You see that in all sorts of cancers, and I've only described 2 of them, we now have the impressive achievement of being able to write their molecular biography at the DNA level and the protein level as well, because DNA encodes proteins. So now you can start talking about targeted therapies, blocking 1 or more of those steps. We'll talk about this later on in the lecture. I mentioned that sometimes there aren't just DNA changes that are permanent, they're epigenetic changes. And epigenetic changes are changes in gene expression. So here's an example with the P16 gene. In the P16 gene that's mutated in colorectal cancer, here's the promoter. The promoter is usually turned on in P16, so it's a tumor suppressor. The gene is expressed, and it blocks cell division. However, when methylation happens, when methyl groups are put on the cytosines of the promoter, they attract a bunch of proteins that glop onto DNA and stop it from being expressed. And that's what happens in cancer, in the lung cancer. The P16 gets turned off, and so you don't get expression, and so you don't have this, and you don't have any protein being made of P16, and so you can't block cell division, and cell division then proceeds. So there are both genetic and epigenetic changes that can happen, and these can be detected by modern genome sequencing techniques. So given all these molecular descriptions, given everything we seem to know about cancer, how can we go about looking at individual patients for these changes to make diagnoses and to make more detailed descriptions? I've described molecular descriptions based on tumor type. I've said, in the colorectal cancer, the following events happen. In lung cancer, the following events happen. But are all tumors the same? Are all colorectal cancers the same? Well, at the cell level, when you look at the cells, they're not. But at the DNA level, they're not either. There are probably many different mutations in genes and many different mutations in different people. And these differences lead to differences in clinical course and response to treatment. So to try to individualize therapy, to try to do target therapy for people's own genetic problems, we have something called the Cancer Genome Project to try to describe these things. And the idea of the Cancer Genome Project is the following. Every tumor is different. Every cancer patient is different. The goals of this project are to identify changes in the genomes of tumors that drive the progression of cancer. That is the important mutations. By doing that, we can identify new targets for therapy, and we can select drugs for therapy based on the genomics of that particular tumor. Many changes in DNA that happen in tumors are passengers. They're just DNA. They may be even in oncogenes or tumor suppressor genes, but they're not driving the cancer. Okay, we want to know which are the important ones that are driving the cancer. The goals of the Cancer Genome Project are to obtain a comprehensive description of changes in the DNA genome, the genome, of gene expression, remember the epigenetic changes I just described, as well. We're trying to do it in 50 different tumor types and subtypes, which are the most important in terms of numbers all around the globe. Give you an example, initially they found 200,000 genetic changes that they hadn't seen before in 600,000 tumors, just looking casually and not sequencing the whole genome. So many genomes from tumors have been sequenced. If I was standing here a year ago, I would say, well, we're just starting. We haven't sequenced many of them. As I stand here now, we've sequenced thousands. As you watch the course, they will have sequenced thousands more. A related effort to looking at the DNA sequence is to test for drug sensitivity. Different drugs work on different tumors, as we're going to see. And the idea here is to take, as we're doing the sequencing of DNA, take cells and biopsies from tumors and screen for drug sensitivity in the lab. I do this all the time in my own research. You take some cells from a patient, you put it in a laboratory dish, and you give it some drug and you say, are you dividing or not? Grow or not, right? And by doing this, we may uncover new mutations that involve resistance to chemotherapy or sensitivity to chemotherapy. To give you an example, you remember Gleevec, the drug that's used for chronic myelogenous leukemia is treating the basketball player in the first example I gave in the course. It's turning out that some people are resistant. They're developing a target, the gene that codes for the target for this drug, which is the gene from that chromosome translocation, is mutating itself as the tumor develops. And as it mutates, it's no longer a target for Gleevec as was developed, and so we now have second and third generation of Gleevecs. And that's all going to come from the drug sensitivity testing of multiple tumors. There are many genome projects all around the world. For example, in one of the efforts, different tumors have been assigned to different countries' scientists. I have to emphasize, while I'm an American, I'm proud of American scientists, etc., science is international. And the same methods are used in China and in India and in Switzerland and in Great Britain as they're used scientifically in the United States. And they're no better and no worse, and they're very good. And so people are cooperating a great deal in this effort. For example, in this effort, in this instance, Australia is going to do ovarian cancer, prostate cancer in Canada for the genome sequencing, stomach in China, European Union, kidney, etc. To give you one example, to zero in on pancreatic cancers being done in Australia, the goal is to sequence 375 patients. It's being met as we speak. Sequence the tumor DNA, determine gene expression and epigenetic changes. It sounds nice to say, but it's very sophisticated and involves a lot of work by a lot of people. So they'll do the sequencing and analysis at an institution in Australia called the Australian Institute for Molecular Bioscience in Brisbane. And the clinical work is being collaborated between people in 3 countries, between Australians, Italians, and Americans, to relate these things. Now, what is the results so far of the anticipated results that we get? Well, first of all, what we're finding is that tumors have a lot of mutations. I've actually seen it said, and I don't know whether this is true or not, that every cigarette one smokes for an entire smoke of one cigarette causes a mutation in the lung. That's a lot of mutations when you think about it. So there are a lot of mutations out there. Secondly, not all mutations are important to cancer. Some mutations are not in DNA that's involved with oncogenes or tumor suppressor genes, and even some of them that are involved in oncogenes and tumor suppressor genes are not the drivers that are pushing the tumor to divide. Others are just there. And identifying which are the drivers and passengers is a major challenge. Third, we're finding tumors are heterogeneous, even among themselves, not between patients, but heterogeneous. We knew this already. We knew that different mutations accumulate in different cancers. We know, as I mentioned earlier on in the course, some cells in the tumor might be resistant to chemotherapy. Some cells are stem cells that are very tumor bearing as the tumor grows, and others, even the bulk of the tumor, some of the cells are not tumor cells anymore per se. They've lost the ability to form tumor. This presents, again, an enormous challenge to therapy moving toward a molecular identification. So this is really our goal, isn't it? It's to identify each tumor in each person specifically for its driver mutations and then design therapy based on that molecular description. This is very different than the way cancer is currently diagnosed. Now, when you go for a diagnosis, it's done by location. Where is the tumor cell? In the breast, for example. And pathology. Look through the microscope and say, oh, that's a ductal carcinoma of the breast, stage something or other, etc., and this is how we treat it. So to go to a more precise level is going to be an amazing achievement, and it's going to happen, and it's happening now. The outcomes of this knowledge are going to be better classification of tumors to predict, for prognosis, that is, how do we proceed, and response to therapy, of which drugs will work. And second, of course, to develop new targeted therapies. There's going to be a lot of genetic testing going on. Genetic testing of tumors, genetic testing of people. And so we have to look at when genetic testing is done of people, even of cancer, what do we do with that information? There are ethical and legal considerations to all of this. Recall the last lecture I described a woman named Ellen Remember, she was seeing her doctor. There she is, seeing her doctor. She's not about breast cancer. She's seeing her doctor and asking about getting tested for the breast cancer gene. And we know now what the gene is. It's the BRCA1 gene. And the gene was tested and sequenced. And the sequencing was done by a company in Utah, Myriad, who holds a patent for this. We'll talk about that in a couple of minutes. And the testing gave the result that she has a mutation, and she chose to have her breasts and ovaries removed. So that could then, obviously, if you have the offending cells gone, you can't get the cancer. There are going to be many more of these tests developed as time goes on. Colon cancer is now already there, etc. So many colon cancer, many things can be developed. Now, some of these tests may be offered to the public directly. Here is an example I'm showing now of, it says, if you can see this, it says a BRCA analysis and it's actually from Myriad. This is an ad that was placed experimentally in a couple of markets in the mid-2000s for about 6 or 8 months. And they are direct-to-consumer advertising saying, you worried about breast cancer? Do you have members of your family with breast cancer? Go to your doctor and ask for this test, because you can only get it right now by prescription. There was a howl of controversy about this in the medical community. And the reason was the average person is not necessarily intellectually equipped with the information to make a decision that they are at risk for having this test. The test costs several $1,000s. Costs a couple of $1,000s to sequence all the DNA. And are you willing to spend a couple of $1,000s? It might not be covered by insurance if you don't have risk factors, if you don't have first-degree relatives who have breast cancer and young age and multiple tumors and bilateral tumors. So if you're not at risk, if you're a 60-year-old woman who has no risk factors, and yet you're still worried about this for whatever reason, you're maybe taken advantage of here because the risk is quite low that you have this mutation. Because BRCA1 gene is only a very low percentage of inherited cancer, and it's unlikely that a person who's 60 has inherited cancer. So this was very controversial, and they withdrew the advertising. What's available now, as I speak, are susceptibility information using the linkage, as I described earlier on with Marie Claire King, of DNA linked to base changes, the small base changes in DNA linked to a phenotype. So here, for example, is me. Here am I. This is one of the companies I sent my DNA to. Actually, I sent my saliva to it. And the disease risk that they gave me information on is called squamous cell carcinoma. It's a skin cancer. And what they said is David Sadova has, at this particular location in his genome, 2 Gs adjacent to one another. And that's an average risk for squamous cell carcinoma. If I had 2 Cs adjacent, I would have a different risk. How do they know this? They analyzed genomes of 1,000 people with squamous cell carcinoma. Okay, it's a cohort study. A case control study, cases, 1,000 people, 3,000 controls, looked at this mutation or looked at this sequence and said, who's got the mutation? And they said, well, okay, if you have a G here, you don't have it, and the controls in the cases had the 2 Cs, right? The G right beside one another. So the risk is about 1 in 500 per year. I have the G. I'm an average risk. It's not lethal. Here's the problem with doing this. Can the public understand this? This is direct-to-consumer. Anyone can order this. So direct-to-consumer testing is very controversial, and here are some of the arguments for and against. By and large, the direct-to-consumer testing that people say, let's do it, say, we're educating patients, we're providing information they can use. The people saying opposed say, you're not educating people at all, you're just selling a product and scaring them. You need to be high literacy to read this stuff. In the thing I just showed you of my own DNA sequence, it's written by a fantastic human geneticist has written this. She's got an MD. I thought it was great. I showed it to some neighbors who have no education in science and had not seen teaching company course on DNA and genetics, and they had no idea what was going on. This is empowering people, say the people who are opposed, who are in favor of it. It's giving people information they can use. On the other hand, it's eroding the patient-provider relationship. People are going in saying, I know what's wrong with me, cure me, right? On the one hand, it's saying, well, it's a great model because it will be patient-centered. On the other hand, we're going to increase healthcare costs because people will be worried and they'll want this kind of care. So there are arguments pro and con. Which brings me to some discussion of ethics. There's the issue of autonomy. There's the issue of maleficence. Don't do anything wrong, maleficence. Non-maleficence, right? Where you're not doing any harm, and beneficence and justice, to do the right thing. What do I mean by that? Let's look at Ellen. First, autonomy. Ellen got the test. She has the right to get a test to find out whether she has the gene, and the right, in case she had the mutation, and the right to make a decision. And she did make a decision on it. She's autonomous. She has the right to know and act freely. Non-maleficence. Don't do any harm, right? What if Ellen got a test, and which she did, and had and has the mutation, and has survivor guilt if she didn't have the mutation, or she gets very upset if she has the mutation? You're creating psychological harm. What if she did nothing? Didn't want to get any more mammograms, she just says, I'm going to live my life, right? She's not acting in a good way toward her own survival. Beneficence and justice. What if she has the mutation and decides not to tell any relatives? Doesn't she have a duty to tell all of her relatives, this mutation is in our family, you should get tested because if they have children, they could pass it on. People worry about discrimination if you have a mutation. In America, there's the GINA, Genetic Information and Non-Discrimination Act, which prohibits discrimination on the basis of genetic information. Most countries and states have these laws now. But they don't prohibit anxiety of having a mutation, getting that information. They don't prohibit the psychological issues of doing. Then there are legal aspects. A gene sequence can be patented. Myriad patented the gene for the breast cancer mutation. And they said, well, we've patented the breast cancer mutation. We're the only people who can do it. The patent is now being challenged. It was challenged by a number of people in the courts. An initial court ruled it's not really, it's not an invention. They said, we take the gene out so it's not the same as it was inside the patient. It's something new. It's not natural. You can't patent something that's natural. So the lawsuit was initially ruled in favor of the people and said, no, this is not a valid patent. DNA cannot be patented. Then a appeals court ruled that that's wrong, and the appeals court said, no, you've got to, this is a legal patent, you can't patent the gene, Myriad can have a monopoly and charge whatever they want. It's now obviously going to be argued in the US Supreme Court, and there's a similar court challenge going on in Australia. So I've described now the molecular biology of cancer. I've described how the biography of the cancer cell works. It's an amazing achievement. We've peered inside cancer cells for what really is a deep understanding of what's going on. From now, we're going to go back in the clinic, and I'm going to describe the way cancer is treated and the science behind cancer therapy. Thank you.